The neurobiological architecture of love and attachment. The experience of falling in love and forming deep emotional bonds with another person involves some of the most powerful and transformative neurobiological processes known to science. Modern neuroscience has begun to unveil the intricate brain circuits, neurotransmitter systems, and hormonal mechanisms that underlie human pair bonding, revealing love to be not merely a cultural construct or poetic metaphor, but a sophisticated biological phenomenon rooted in ancient neural systems that we share with other pair bonding species. The neurobiological story of pair bonding begins with the recognition that romantic love appears to activate three distinct but interconnected brain systems. The first system involves sexual attraction and is driven primarily by testosterone and estrogen. This system motivates individuals to seek out potential mating partners and creates the initial physiological arousal associated with sexual desire. The second system encompasses romantic attraction and is characterized by intrusive thinking about a specific individual, euphoria when the relationship is going well, and despair when it faces obstacles. The third system involves attachment and is associated with feelings of calm, security, and emotional union with a long-term partner. At the molecular level, two neuropeptides have emerged as particularly crucial for pair bonding behavior: oxytocin and arginine vasopressin. Often called the love hormone, oxytocin plays a fundamental role in social bonding across many species. Originally known for its role in childbirth and lactation, oxytocin has been discovered to be essential for the formation of social attachments in general. Research using prairie voles, one of the few naturally monogamous mammalian species, has shown that oxytocin released during mating facilitates the formation of partner preferences that can last a lifetime. Oxytocin acts on specific brain regions that are part of the reward and reinforcement circuitry. When prairie voles mate, oxytocin is released in areas such as the nucleus accumbens and ventral striatum, where it binds to oxytocin receptors and creates positive associations with the partner. This process essentially hijacks the brain's reward system, making the presence of the specific partner inherently rewarding and motivating behaviors that maintain proximity and contact. Species differences in the distribution and density of oxytocin receptors appear to determine whether a species will be monogamous or polygamous. Vasopressin plays a complementary but distinct role in pair bonding, particularly in males. While oxytocin tends to be more important for female bonding behavior, vasopressin appears to be crucial for male attachment and territorial behavior associated with pair bonds. Male prairie voles that receive vasopressin directly into their brains will form partner preferences even without mating, while blocking vasopressin prevents pair bond formation. The distribution of vasopressin receptors in the brain, particularly in regions like the ventral pallidum, appears to be a key determinant of monogamous versus polygamous mating strategies. The dopamine system provides another critical component of the pair bonding circuitry. Dopamine is widely known as the neurotransmitter associated with reward and motivation, and its release in response to pair bonding activities helps explain why romantic relationships can become so compelling and even addictive. Early stages of romantic love are associated with elevated dopamine activity in the brain's reward centers, creating the euphoria, focused attention, and obsessive thinking characteristic of new relationships. Over time, as relationships mature, dopamine responses may shift to promote attachment and long-term bonding rather than just initial attraction. Brain imaging studies in humans have provided remarkable insights into the neural activity associated with romantic love and attachment. When individuals who report being intensely in love view photographs of their romantic partners, specific brain regions show increased activation. These areas include the ventral tegmental area and caudate nucleus, both part of the brain's reward system, as well as regions associated with attention, motivation, and goal-directed behavior. The pattern of brain activation during romantic love shares similarities with other forms of addiction, suggesting that pair bonding may literally be a form of natural addiction to another person. The temporal dynamics of these neurobiological systems help explain how relationships typically develop over time. The initial phase of romantic attraction is characterized by high levels of dopamine, norepinephrine, and phenylethylamine, creating the excitement, energy, and focused attention of new love. This phase is often followed by increased oxytocin and vasopressin activity as physical intimacy and emotional bonding deepen. Eventually, the neurochemistry may shift towards systems involving endogenous opioids and GABA, which promote the calm, comfortable attachment characteristic of long-term relationships. Individual differences in neurobiological functioning help explain why people vary so dramatically in their capacity for pair bonding and relationship satisfaction. Genetic variations in oxytocin and vasopressin receptor genes have been associated with differences in attachment styles, relationship quality, and even divorce rates in humans. Some individuals appear to have genetic variants that make them more responsive to the bonding effects of these neuropeptides, while others may be less sensitive to their influence. The stress response system also plays a crucial role in pair bonding dynamics. Cortisol and other stress hormones can interfere with oxytocin and vasopressin signaling, making it more difficult to form and maintain strong emotional bonds during periods of high stress. Conversely, the presence of a supportive partner can buffer stress responses and promote more effective coping with life challenges. This creates a positive feedback loop where good relationships promote better stress management, which in turn supports stronger relationships. The mirror neuron system contributes to pair bonding by enabling partners to understand and empathize with each other's emotional experiences. These neurons fire both when an individual performs an action and when they observe their partner performing the same action, creating a neurobiological basis for emotional synchrony and mutual understanding. This system may be particularly important for the emotional attunement and empathy that characterize successful long-term relationships. Recent research has begun to explore how modern lifestyle factors affect the neurobiological systems underlying pair bonding. Chronic stress, sleep deprivation, substance use, and digital technology use can all influence neurotransmitter function and hormone levels in ways that may interfere with relationship formation and maintenance. Understanding these effects has important implications for helping couples maintain strong bonds in contemporary environments that may be poorly matched to our evolved pair bonding systems. The study of attachment disorders and relationship difficulties has provided additional insights into pair bonding neurobiology. Individuals with histories of trauma or neglect often show altered oxytocin and vasopressin functioning, which may contribute to difficulties in forming secure attachments. Therapeutic interventions that target these neurobiological systems, including certain medications and behavioral therapies, show promise for helping people develop healthier relationship patterns. Gender differences in pair bonding neurobiology help explain some of the documented differences between men and women in relationship behavior and preferences. While both sexes rely on oxytocin and vasopressin for bonding, the relative importance of these systems and their interaction with other neurotransmitters appears to differ between males and females. These differences may contribute to documented variations in relationship priorities, communication styles, and responses to relationship stress. The neurobiological systems underlying pair bonding continue to function throughout the lifespan, though they may be modified by aging processes. Research suggests that older couples who maintain strong relationships show continued activation of reward and attachment systems when viewing their partners, indicating that the neurobiological basis of love can persist across many decades. This finding provides neurobiological support for the possibility of lifelong pair bonds and may help explain why some couples report growing closer rather than more distant over time. The implications of understanding pair bonding neurobiology extend far beyond academic interest. This knowledge is informing the development of new therapeutic approaches for relationship problems, helping couples understand the biological basis of their emotional experiences, and providing insights into how to maintain strong relationships throughout life. As our understanding of these systems continues to develop, we can expect even more sophisticated approaches to supporting human pair bonding and addressing the relationship challenges that cause so much suffering in contemporary society.